On today's video, we'll be explaining the mysterious chaos engine that I've been speaking about that powers and is at the heart of my theory. Hi, my name is Eduardo Hernandez. I am the creator and author of Emergent Chemical Evolution, the newest theory on the origin of life that solves every step of the problem, beginning from the first molecules until modern cell. So let's get to it. The chaos engine is what I've named the environmental conditions that emerged and coalesced to start life. According to my theory, and I've explained this in other videos, which I'll link to in the descriptions below, the atmosphere was laden with carbon dioxide, therefore creating pH gradients, lower pH, more acidic at the surface, and lower going, and gravity creating currents in the ocean, and the ocean's current being so strong that they can take any material and pass it through these different pH gradients, especially closer to the surface. So that being said, I'm sorry, I get flustered. Remember, this is raw. It'll be edited out later, so please bear with me. That creates a perfect situation for the emergent chemical evolution, which is why I named my theory that, of molecules progressing through these pH gradients and therefore changing and setting the stage for the emergence of life. Now, exactly how that happens is what this video is about. This video we're going to be talking about right now. I'll just start. Go ahead. One of the main ingredients of life, the one type of molecule that does all the work for life, are amino acids. Amino acids are the proteins. The proteins are the enzymes. The enzymes work by transforming chemical compounds into other compounds. Usually, and for the most part, what life starts to transform to create the biomolecules it needs are amino acids. Amino acids in almost all metabolic activity is the initiating molecule. The one that's most important to all life is the creation of RNA. Life cannot translate the DNA code into proteins without RNA. RNA is the template that ribosomes, which are also composed of amino acids and proteins, and RNAs, which is the main catalytic component of the ribosomes, which cleaves the amino acid from the transfer RNA and unites it with the chain of protein. Again, RNA is the most fundamental molecule necessary for life. So, that being said, like I said, my primary my ideal example or life form is the E. coli. Again, we're going to start with the E. coli because it's one of the simplest cells known to man. It doesn't have a nucleus. It only has one chromosome. It doesn't have mitochondria. And the way it creates RNA is very important to me. Well, not just important to me, of course, important to the E. coli, but it's a very important step. And it also is an ideal model, or example, I should say, of emergent chemical evolution being a clear method for that to have initiated. The E. coli uses 12 smaller polypeptides. Polypeptides are small chains of amino acids that 12 of them have to get together, of course, in a specific pH, in a solution of a specific pH that allows each polyamino acid to take a certain conformation and allow them to form together in a jigsaw puzzle, and then take an amino acid and transform that amino acid into a precursor, a molecule before the RNA molecule. So this is the initiating step to create RNA. Now, why, after billions of years, does the E. coli, or has not the E. coli, evolved a more efficient, a more a system with less components to do the same job? Because, just like I've said before, life, once it solves a problem, rarely changes from using any other mechanism. It'll either make it better or make it worse, depending on evolution and other forces. But once it grabs onto a problem, it sticks to that problem. And it will rarely ever change. And that is clearly the case because 99.9% of all animals that have ever existed have gone extinct. And that's because once life solves a problem for an organism within its niche, if that niche changes, evolution cannot change fast enough. In most cases, not all. In most cases, it cannot change fast enough to allow these animals to evolve and get accustomed to another niche. So again, I mean, E. coli is a prime example. It, after billions of years, has not formed or evolved a mitochondria. It has not evolved a nucleus. It has not evolved a more complex mechanism that might be simpler to create RNA. But how did that system to create RNA begin in the first place? 12 smaller polypeptides uniting to initiate RNA. To me, the most obvious reason is that my theory is correct. My theory shows, and well, actually, that's what I need to be talking about. Actually, let's get back to that. But that is a prime example of why I believe my theory is correct. And let's get back to the original reason for this video. So, all that being said about amino acids and the importance of amino acids in metabolic activities, and the simplest life form that we know of utilizing small polypeptides to initiate one of the most important activities of life, the creation of RNA molecules, shows that the basis, that the result of the mechanism of the chaos engine that is fundamental to my theory is a perfect explanation for why we find that in E. coli and other simpler organisms. All right, so again, with the situation being as it is, carbon dioxide laden atmosphere creating gradients in the ocean, the gravity creating currents to grab the the material of the ocean and pass it through the gradients. And again, the heavy bombardment was responsible for the volatile compounds of our Earth to repopulate the Earth after the Earth melted and became a big fireball, and initially irradiated into space the volatile materials, the light gases, water, the lighter elements were burned off. So after the Earth cooled down and then the heavy bombardment came, the heavy bombardment returned those volatile materials. This is not my speculation. This is one of the main theories of astronomy. And the evidence, I reference the articles in my book that explains that the inventory was replenished by the heavy bombardment, which brought back the most important thing that we have on this planet, water. Drop by drop, over millions of years and millions upon millions of impacts of these meteorites, which were laden with water and volatile materials. Among those, the organic materials, carbon dioxide, and amino acids. The most important source of amino acids was, as far as I'm concerned, the meteorites. A large portion of the carbonaceous meteorites contain up to 80 different types of amino acids. Or 70. 80, 70. I really don't remember. I think it was 70. 70 because I remember it was the Murchison meteorite was found to have amino acids in 1969, which was the year before my birth, and it correlated with the 70 different types of amino acids, at least in the articles I've read. Where was I? I'm sorry. So let's get back. So once the heavy bombardment began and replenished the water, basically supplied the water for our oceans, because drop by drop, I mean, can you imagine the number of impacts necessary to bring back the oceans? I mean, to bring the oceans to this planet. And at the same time, they were laden with amino acids. So it's virtually impossible for the oceans to have existed without being saturated with amino acids and other carbon and other material, other elements carried by these meteorites. The one thing is, amino acids are soluble in water. I had someone comment on a previous video about one particular amino acid not being soluble. Now, it is the least soluble amino acid among the amino acids used by life, but within 1 liter of water, 11 grams of the material is soluble in water. Now, albeit this is not 1 mole. The molarity and solubility is based on the overall molecular weight of the compound, and that has to do with the number of molecules, Avogadro's number, which gives you the weight of the molecule and how much grams you need to have in order to have Avogadro's number of molecules. Now, that being said, the molarity of this amino acid is very, very low, meaning that the number of grams to have a whole mole of this amino acid compared to what's soluble in the water is very tiny. But that gives millions of different molecules available in the water of that particular amino acid. It's not a large portion of the molar weight of the compound. but it's still a significant amount. Remember, life on Earth operates at the molecular level. If life has one molecule, it's going to do something with that molecule. So you don't need the whole or a large portion of the molar weight of that compound to react or to make things happen. When you're dealing with us in chemical laboratories, yes, that becomes important because we can't work at the molecular level. We need to work at the molar and proportions of the molar weight. So if we can't get enough molar weight of a certain compound, it becomes difficult for us to cause chemical reactions and to control chemical reactions in laboratory settings. But life didn't need that. It needs one molecule with one other molecule to work. So that is not a hindrance to my theory. It's not a hindrance to life being able to start in water. So that being said, all amino acids were available in the water at the time of the initiation of the chaos engine. The initiation of the chaos engines occurred when there was enough water to create a pH gradient and enough water for gravity to create currents within that water to move the contents of that water between the pH gradients. Why is that important? That is the key mechanism, the key source of energy as well, to power the origin of life. This mechanism continuously cycles this material since the beginning of the oceans. It continuously, day in, day out, second after second, over billions of years, you had the currents moving materials, the compounds within the oceans through those gradients, non-stop. Nothing was going to be able to stop it. It still doesn't stop it now. Well, of course, we have life forms now, but those materials, especially water-soluble materials, would be moving in and out and through those pH gradients. That being said, amino acids is very important. The amino acid is a cation. It is an ion that has a positive and a negative side. And that is the reason why it bonds and forms long chains, because it has that ability. It's an ion that has a negative and positive side, and another amino acid's negative side can attach to another amino acid's positive side, and there it goes, there on and on and on. Now, the important thing about the 20 amino acids that are used by life, and what a lot of origin of life researchers consider a hindrance, is that the activation of certain amino acids is very, very different. You have certain amino acids that are only activated at a pH of 3 or 4, and the other side of the same amino acid would be activated at a much different pH level, a more basic, maybe 9 or 8. So they were like, how could that be accomplished if these compounds are going to be found in water? But again, they never looked at how that water was going to be affected by the atmosphere and the carbon dioxide, creating pH gradients. One of the most popular origin of life theories now is of organic material being found in small puddles that go through drying and wetting cycles. What happens when it dries and no water comes back? Or it dries and it gets buried by other material and it doesn't come back. That is not first of all, there's no evidence. Most of the evidence points to the Earth being completely covered in water for a very long time. and those drying and evaporating cycles would have had to have happened at the beginning of the formation of the oceans. And once the oceans formed, if viable molecules were not initiated or were not preserved, that would be the end of the chance for those cycles to return. They would have been completely covered in water, and there would be the end of drying and evaporating cycles. So I find it untenable. First of all, because to me that type of system is not conducive to attaching amino acids or creating RNA in a sustainable environment. Because I believe if you dry RNA, and depending on the temperature, they can decompose. So I'm not sure how tenable that those chemical reactions would have continued to preserve molecules that would initiate life. Anyway, back to my theory, back to the chaos engine. So the activation of these different amino acids, which within one amino acid, each side would have to be activated at a different pH level in order for it to bond to another amino acid, is solved by the pH gradients within the ocean. That same amino acid could be taken closer to the surface where it's more acidic, and one of its side would be activated, and at that point, whatever susceptible amino acid was nearby, which also its negative side, let's assume, its negative side would have been activated at that pH, cause it to bond to the other positive side of the second amino acid, or the first amino acid. So the case being is that at certain pH, the amino acids that would activate at that pH would interact. Be it that one amino acid would have its positive side activated at that pH, and another amino acid's negative side would be activated. They would easily bond at that point. And then as the current continues, it now has one more or several more polyamino acids attached to that same chain. And as it would cycle through, this would continue to happen, and at different pH, different amino acids would attach and elongate the polypeptides. Now remember, this is occurring over every inch of the globe in the ocean. The Earth is millions of square miles on its surface. And remember the molecules, the size of the molecules. Every single millimeter, every single area of those millions of square miles of ocean would have this occurring below its surface. The ocean will continue to churn the currents and continue to move those materials within it through those pH gradients, creating ever longer polypeptides. And again, what controls within modern cells, what controls the metabolic activation of certain reactions? They're coordinated by the pH of the cytoplasm. The cytoplasm is the medium where is close to the contents of oceans. The cytoplasm would contain these amino acids as enzymes and as starting substrates, and would react only at a certain pH. So how do you mimic or control that before life began? Again, the gradient of the ocean would be a perfect substitute for that. Whereas certain reaction will occur in certain pH in the ocean, so that certain reactions would only occur at a certain pH, because at that pH, certain polyamino acids would become rigid or take a conformation and then only interact with polyamino acids, polypeptides that would link with it at that pH. I mean, if you look at the cytoplasm now, you have tons of different amino acids, tons of different enzymes, tons of different compounds that are all mixed in together. The cell wall can only isolate a certain number of molecules. It cannot isolate every single molecule for every single metabolic reaction to occur within the cell. What does happen is the selectivity, the lock and key reactions of amino acids, of enzymes, would allow only the right amino acid or polypeptide to link, and then within the cytoplasm cause that metabolic reaction to occur. So there's no reason we wouldn't expect this to occur in an open environment that's conducive for reactions to occur. Again, the pH would be variable. Certain metabolic activity would not occur at all pH levels. So within these cycles, you would start to get a type of synchronicity where certain reactions would only occur at certain pH level, and then the cycle will continue. And then you will get components that would mimic enzymes today. I mean, the thing is, and this is going to have to be a whole nother video, is that you have, in different life forms, you have different life forms doing the same metabolic activity in a different manner. What's called homolog proteins. They're proteins that do the same reaction in E. coli are different than the proteins that do it in humans. Now, in E. coli, they are made of smaller polypeptides that unite and take the shape of an enzyme and cause that excuse me, cause that metabolic reaction to occur. In humans, we have a much more advanced enzyme system where it's large, one single chain polypeptide, that mega polypeptide that forms, takes a certain conformation at a certain pH, meaning it will become more rigid and engulf the substrate that it needs to react and cause that chemical reaction to occur. Now, the difference between the large enzyme in humans and the small puzzled together enzyme in E. coli is that, of course, they're made of different amounts of polypeptides. This is a single large polypeptide. The E. coli's are small, multiple polypeptides uniting to form a larger enzyme to do the same reaction. A lot of times what we see is that the overall shape of the enzyme of 12 pieces compared to the one of one large one is almost the same. A little bulges more here, bulges less there, but basically they're the same. And when we look at the activating center, the active site of each enzyme, you'll find the same amino acids in the same position within that active region, meaning that the only thing that should stay the same is the amino acids within the active site, and that the other amino acids of the protein, of the enzyme, are only there for structural purposes to maintain and to create the lock and key activation of and selectivity of those enzymes. So that being said, let's go back to the proteins that that create amino acids, RNA. You have proteins that are replicators of RNA. And in the different kingdoms, there are examples of, they're called, well, anyway, that's not important. What's important is that these proteins copy the RNA sequence. There's 20 different types of these proteins that copy RNA sequences. In each kingdom of life, there are multiple versions of that protein that replicates RNA. These different 20 types, again, like E. coli, certain ones are made of multiple pieces that unite to do the job. And in more advanced animals are less modular. They're larger proteins that do the same job. Now the point is that you have 20 different versions of this protein, meaning that you have 20 different types of sequences of amino acids that can unite to do the same job. So that means that the enzyme doesn't have to be exactly like any particular one of those 20. Meaning that the probability of getting one of those proteins is not taking that protein sequence and calculating the probability of that protein sequence coming about. No, there's 20 more types that can come about. And according to my theory, and according to some of the calculations I did, and that's going to be a whole nother video, there's multiple ways, millions of different sequences that can come and create that same enzyme or that same protein to do that same job. So these calculations that they're doing that only one type of protein has this probability of ever coming out in nature is wrong. I'm sorry. I totally disagree. I don't know if they don't study biology, but it's shown that that's not true. We have 20 different examples of RNA replicating proteins that have different sequences. The difference is more than 45% different. These sequences from one end to the other. The difference in sequence is up to 45%.